Cystic fibrosis, or CF, is the most common genetically inherited chronic illness affecting Australians today. The disease is characterised by genetic mutations resulting in defects in the CFTR protein. Excessive and thick mucus in the lungs causes most of the symptoms, which include wheezing, breathlessness, and chronic infections within the lung. The average life expectancy of individuals with CF is still about 37 years old. 70% of CF patients have a class 2 delta F508 mutation in the CFTR gene, which encodes for an important chloride channel. The mutation causes the synthesized protein to become trapped in the endoplasmic reticulum and thus cannot be expressed on the cell membrane. Water movement onto the surface of airways heavily depends upon the osmotic gradient created by the movement of chloride ions through the CFTR. With defects in CFTR, there is insufficient water secreted onto this surface, and so the mucus becomes thick and sticky. Current treatment options focus on clearing airway obstruction with bronchodilators and inhaled hypertonic saline. Our group has a pioneered novel approach to treat this crippling disease. Retention of viscous mucus in the airways leads to both reduced pulmonary function and exacerbations of infection. Mucus impedes inhalant diffusion across airway epithelium. To overcome this obstacle, our nebulized product requires DNase to thin the mucus. Pulmozyme contains recombinant human deoxyribonuclease 1. In vitro, this hydrolyzes DNA in CF sputum and reduces sputum viscosity. Combining pulmozyme with our nebulized product, targeting the chloride channel 2 protein, is expected to optimize therapeutic efficiency. Thus, use of myolytics facilitates the absorption of our combined therapy drugs into the airways, increasing mucus clearance. The chloride channel protein 2, or CLCN2, acts to mediate chloride efflux and consequent water outflow into the intraluminal space. This channel is present within the gut and the lungs. Constipation occurs in CF patients due to the lack of water in the gut and is treated by taking 25 mg of lubiprostone twice a day. Lubiprostone is a local selective CLCN2 activator and works by stimulating these chloride channels to increase intraluminal chloride efflux and water secretion without altering the sodium and potassium concentrations, thus relieving constipation. Our idea is to re-engineer this drug to an aerosolized form for local absorption across airway epithelium. Mucus becomes even thinner and less viscous, increasing mucus clearance, reducing the risk of respiratory tract exacerbations. We will upregulate the expression of CLCN2 in the airway epithelial cells to further enhance mucus clearance. This is done by the CRISPR-Cas system, a gene therapy focusing on modifying targeted genome sequence. The way it works is that a single guided RNA Cas9 complex induces DNA double-stranded breaks at the desired genome sequence, where it gets replaced with previously designed templates. So briefly, we will be able to transfect the gene sequence for CLCN2 along with an effective promoter to the respiratory epithelial cells. Though 5% of pulmozyme-treated patients develop antibodies to pulmozyme was still able to show 8% increases in mean forced expiratory volume and 34% reduced risk of respiratory tract exacerbations. If patients fail to routinely inhale pulmozyme, then chloride channels may reduce drug response and mucus may reaccumulate. Further, pulmozyme is ineffective for only a subset of CF patients who have a vital force capacity less than 40%. In vitro, lubiprostones are rapidly metabolized on airway epithelium and have a quick onset of action. Hence, they should have no or minimal drug-to-drug interactions. Clinical trials reported that 29% of lubiprostone patients had a dose-dependent adverse drug reaction of nausea. 12% of lubiprostone-treated patients had diarrhea, and about 2% who experienced severe diarrhea discontinued lubiprostone treatment. CRISPR depends highly on the detection of specific gene sequences. However, mutations within sequences in the CLCN2 genes can occur and will not be detected by already designed probes trialed in this study. This may lead to errors in genome editing, and scientists may refer back to conventional diagnostics. Our combined therapy has demonstrated a clear line of research that has widespread implications on treating cystic fibrosis. By combining already existing treatments for cystic fibrosis, such as lubiprostone, and newer scientific methods such as CRISPR, we hope to make cystic fibrosis a disease of the past. Currently, the future of our treatment rests in your hands, and with your support, we can make this therapy become a reality for all those people living with cystic fibrosis. Thank you.